There's a lot going on in our bodies, and it's important that what's inside of our cells is able to know what's going on outside of the cells. And the way that this message is, or these messages, there's lots of different messages are conveyed, is through signal transduction or biosignaling. Basically, there are four main types of such signal transducers that we talk about. And that sounds like a really scary word, but they're proteins basically involved in helping relay that message. These four main types we're going to talk about are going to be G protein coupled receptors or GPCRs. There's a lot of different types of them, but we're going to be talking mainly about the beta adrenergic receptor, which is sometimes see abbreviated beta AR, and the glucagon receptor. And I know it sounds like two different things, but they actually act through the same pathway once you get past that initial GPCR. There's also receptor enzymes, and so our classic ones of these are going to be our receptor tyrosine kinases or our RTKs. We'll talk about the insulin receptor. It's a kind of classic example of one of these RTKs. There's also other RTKs involved in a lot of growth factor signaling. And so both of these can be common sites of mutations that lead to excessive cell growth. And so cancer is often associated with mutations in signaling pathways. And many of these pathways are involving these RTKs and the GPCRs. Both of these have in common that they are membrane-bound proteins that bind to a ligand. So they bind to that extracellular signal. We'll call this the primary messenger, this extracellular signal. And then the signal doesn't actually come in, that signaling molecule doesn't actually come in. So that glucagon, that adrenaline, that insulin, it's not actually coming into the cell. Instead, it's binding to the receptor in the cell. The receptor is then going to undergo a conformational change. It's going to change shape in response to that ligand binding, that signal binding, that messenger binding, that primary messenger binding, and then it's going to change shape and cause things to happen inside of the cells. In the case of our G protein coupled receptors, we'll go much more into these, but basically you're going to have the ligand bind. It activates this G protein that's bound to that G protein coupled receptor. So you have the receptor, which is the GPCR, binds to the ligand, and then that G protein that is bound to the receptor is only bound temporarily. And once this receptor gets activated, then it activates that G protein, and that G protein goes away and it activates other things. And this will lead to the production typically of other secondary messengers inside the cell, so small molecules like cyclic AMP that are then going to be able to carry out big effects. And this is going to allow us to amplify that signal inside of cells. Our RTKs, here what's going to happen is that binding of that ligand actually activates the kinase portion of the receptor tyrosine kinase. And the kinase is going to phosphorylate things. And one of the things it phosphorylates is typically another kinase. And then that kinase phosphorylates another kinase, and you get this cascade of phosphorylation. And that cascade of phosphorylation leads to other things happening, including controlling the transcription of genes. And so actually going and making mRNA from genes that you can then make protein from in order to get proteins made in response to an extracellular signal. I know that I'm saying, and then a lot of stuff happens. There is a lot of stuff that happens, and it's not just that I don't want to go into all the stuff that happens, but there's so much different stuff that happens. Because in response to the same signal, you can then imagine this is going to activate different pathways in different cells at different times, all these various things depending on what other players are around. And we'll see how sometimes they're going to be scaffolding various proteins. So you'll have big floppy proteins, some with those like IDRs and stuff, and some of them will have domains that say bind to phosphorylated regions. And then this is then going to allow them to hold together proteins and allow this signaling to happen in various ways and efficiently. And then also to be able to kind of remove all that stuff at once when you want that signaling to end. So these are going to be the two main things we're going to talk about. But there's also, I remember I said there were four things. The other two, there's a gated ion channel. So we already talked about these gated ion channels. Remember, those were going to be facilitating diffusion. So it was passive transport, facilitated diffusion. We've got those charged molecules that can't go through the membrane on their own, but if you let them go through a channel, they'll just flow on in. And we don't want these channels open all the time, however, because that would just kill our gradient and then things wouldn't go in. But if we kind of build things up and then in response to a ligand binding, we've got this big gradient ready to go. The ligand binds, the channel opens, the ions flow in, and then the ions can cause things to happen inside of the cells. And finally, we have just kind of the ligand itself is actually coming in. So its receptor is all the way in the nucleus. This is what we typically see with steroid hormone signaling. Remember that steroids were going to be neutral, and so they could go through the membrane and they could go all the way through the nuclear membrane as well into the nucleus and affect the transcription of genes and allow you to make proteins. And so by controlling the transcription at the transcription level, we're able to have kind of a longer effect. We're able to have bigger changes, but those changes are going to be delayed compared to when we're just phosphorylating a protein and voila, we change shape right away. A critical thing is that in this case of our nuclear receptors, our steroid hormones, this is the only time where that messenger is actually going to get into the cells. In the other cases, we're relaying the message through additional messengers or relaying the message inside of the cells through responses to binding to proteins in the membrane of the cells. So keep those things in mind. And now, hopefully, I can convince you before we get into the details that it's really important. So why care about biosignaling? Here's a bit of a teaser about what's coming in our metabolism section. Today, I'm going to introduce you to two of the main hormones we're going to be talking about, insulin and glucagon, which are going to control largely our blood sugar levels so that our glucose levels are ideal. So what I'm showing you here is a graph of a drug called a GLP-1 receptor agonist, or a GLP-1RA. You might have heard of these. You've probably heard of these. You've seen those commercials for things like Ozempic, and you're seeing how the number of published articles of them has gone zooped up. Lots and lots of articles on them, lots of trials, all these various things, and lots of money to be made. You've probably heard that these are like weight loss drugs or they can be used to treat diabetes, but what actually are these? GLP-1 is glucagon-like peptide 1. It's secreted by the intestines and it acts through GLP receptors. So these GLP-1Rs, and those are GPCRs, our good old GPCRs. So we have lots of different GPCRs, and there are GPCRs that respond to the binding of this GLP-1. This is going to increase insulin production. Insulin is going to say, let glucose in. More glucose is therefore going to be taken into cells. And that insulin, remember, was acting through RTK signaling. So through GPCRs, we're able to increase insulin production. The insulin is then going to act through RTKs, and that's going to have the effect of decreasing glucagon production. And glucagon, remember, was going to act through another GPCR. And so as a result of that, less glucose is going to get made. GLP-1R agonists, like Ozempic, serve as a treatment for type 2 diabetes and obesity. What's this word agonist that I'm talking about? An agonist is going to be a ligand that binds to a receptor and activates it. This is the opposite of an antagonist, which is going to bind to a receptor and prevent the activation. One thing that I want you to take away from today's lesson is that we're going to be seeing a lot of diagrams and a lot of things with arrows. And it's important that you get comfortable with seeing things like arrows indicating activation, often green arrows. And then you might have something like this where you have a red line with a vertical line at the end. I don't know what you would call this because kind of horizontal T. And this was going to indicate that you're inactivating something, you're blocking something. So I'm not caring that you know all the details about all these pathways. Instead, I want you to get familiar with some of the depictions of these pathways. I want to empower you to be able to read diagrams. That's kind of one of the main goals of this lesson. One of the other main goals is to introduce you to some of these players that we're going to be seeing throughout the metabolism section of our course. Note that we're talking here about GLP-1, which is glucagon-like peptide 1, not glucagon. So glucagon actually has an opposite effect. So we saw that GLP-1 was going to decrease glucagon production, and that was going to lead to less glucose being made. Because glucagon is a hormone that says all the glucose is gone, and so let's go and let's make some glucose. And we'll see how that making of glucose is going to be happening mainly in your liver. But glucagon and GLP-1 are actually small peptides that are made from the same precursor, this proglucagon. This can get chopped up in various ways in various tissues. And so in your pancreas, it's chopped in a way that gives you glucagon. And then in your gut and your brain, it's chopped in a way that gives you various things, including this GLP-1. We're going to talk a lot more about glucagon and insulin as we get into metabolism more, but basically these are important hormones for controlling blood sugar. In response to low blood sugar, what's going to happen is that glucagon, this hormone glucagon, is going to get released by pancreatic alpha cells. I like to think of it thinking all the glucose is gone. And so if glucose is gone, what are we going to do? We're going to make glucose anew. So that happens in your liver and kidneys. And we're going to break down glycogen. So our storage form of glucose is glycogen. We've got these long chains of glucose attached to this central protein. And the glucagon is going to promote the breakdown of this to give you individual glucose units. Energy, yay. Okay. Now, insulin, however, is going to say, beta let some glucose in. And so if there's a lot of glucose around, then insulin signaling is going to make it so that that glucose gets let into cells. We'll see how this actually happens in response to receptor tyrosine kinase signaling. And then it's going to signal other things, and it's going to promote the linking together of these glucose units into glycogen. In diabetes, what can happen is that either people don't produce enough insulin, which is the case in type 1 diabetes. Often this is due to autoimmune attack of the pancreas because insulin is made in these pancreatic beta cells. Or in type 2 diabetes, a person doesn't produce a strong enough response to insulin, so they've kind of become desensitized to it. So insulin is saying let the glucose in. In diabetes, the glucose isn't coming in. And our insulin signaling is kind of defective, so let's turn to the other branch. Our other branch is going to be that glucagon. Now, glucagon is going to promote the production of glucose. It's going to promote the release of glucose into the bloodstream, so we need to inhibit it. So diabetes can be treated with insulin, giving them insulin, or inhibiting the glucagon. And when your insulin signaling system is kind of defective, then the glucagon seems like a good avenue. And what we could do is we could go and we can actually take advantage of that GLP-1, that natural signaling molecule that is able to promote the inhibition of the glucagon-based signaling. So this GLP-1 scientists are like, okay, well, can we take that GLP-1 and actually use it as a treatment for diabetes, as a treatment for obesity? Well, the GLP-1 itself wasn't stable enough, but they were able to look to other animals' GLP-1s and to look to the biochemical basis of all the binding and all that great stuff and create these modified versions that are more stable but still have that same effect. And these have had a dramatic effect on the health of a lot of people. And so here's just kind of one teaser as to why this signaling is really important. And although this signaling is going to get really complicated, and you'll see complicated diagrams like this, where the GLP-1 is then influencing the release of insulin, and it's inhibiting the release of glucagon, and you have these different receptors and all these arrows, and oh my gosh, relax. You don't need to memorize anything. You need to be able to interpret diagrams. If you can interpret diagrams, you're good. Because there's so many different pathways that it doesn't make sense to memorize them. Instead, you want to be able to know the fundamentals of these basic types of signaling and then how they can be interconnected in different ways. You're going to see crosstalk between different pathways I'm going to give you a couple of examples of important ones, but those are far from the only ones. So we're going to talk about this glucagon or beta adrenergic receptor. You have a glucagon receptor and an epinephrine or adrenaline receptor. Adrenaline and epinephrine are the same thing. They have different GPCRs because remember the GPCR was the part that actually bound to that ligand, that signal. But then they used the same downstream pathway, this G stimulatory, this GS. And so there's different G proteins, but we're going to focus on this GS one that stimulates adenylate cyclase that produces the second messenger cyclic AMP, so a small molecule inside of the cells that then activates this protein PKA, and then that is going to have effects on the glycogen breakdown and glycogen synthesis. And then insulin is going to act through the insulin receptor, and then that is going to have effects on the glycogen breakdown and glycogen synthesis, as well as a bunch of other things, including the bringing of glucose transporters to the surface of cells. And so insulin and glucagon, we're going to see them over and over again. When you think glucagon, think beta adrenergic receptor. It's not acting through the beta adrenergic receptor, but it's acting through the same downstream pathway. Glucagon is only going to have receptors in certain tissues like your liver. And so your muscles, say, aren't going to respond to glucagon, but they will respond to adrenaline or this epinephrine. And GLP-1 receptor agonists are going to have opposing effects to that glucagon and increase the effects of the insulin. We're going to dive into some of these pathways now. And these are some of the things I want you to keep an eye out for. These are things that you would want in your signaling system and components of signaling systems that we find. And one of the reasons why these pathways are so complicated. They need to have specificity, so you only want them to be binding to the specific thing you want them to respond to. They need to be sensitive, and so they need to be able to bind to that receptor, even if there's not that much of the ligand around. So we need to have a low KD. But that's not all. We want to make sure that even if we have a low level of the signal, that signal then gets amplified inside of the cells. And so we need to take that message that's outside of the cells and amplify it inside of the cells. And these cascades that we'll see are going to be a great way to do that. There are going to be lots of signals coming in, though, and we're going to have to be able to integrate signals that we see from different receptors. And so this is where we get to sort of crosstalk between the different pathways. We need to be able to take a signal from outside and branch off into different responses. We need to be able to diverge the signal once we are at the receptor. And so we'll see that the same receptor can activate different pathways. One of the ways that we can do this is by having modularity. So we have these different proteins that can be scaffolded together in different ways, kind of like Lego pieces, you can hook them up to different things. Signaling is great, but too much signaling, not so great. And so we need to be able to say, okay, we've done enough, let's turn the signal off. And so we need to be able to desensitize or adapt to the signal. And we need to keep that signal local. And so we don't want to make changes all over if all we need to do is to have things nearby. And so if it's raining in Philadelphia, you don't need to bring out your umbrella in California. So localized response. So keep an eye out for how these pathways that we'll see are going to be incorporating these different things. If we want the quickest action, we're going to want to regulate at the level of post-translational modification. The main one we're going to be talking about is phosphorylation. So the protein's already been made and it's folded up nicely to accommodate all of its different amino acid residue side chains. And now you come and you put a phosphate group on them. And so these phosphate groups are typically going to be put on serines or threonines or tyrosines, those amino acids that actually have these hydroxyl groups that can then get phosphorylated. And the proteins that phosphorylate them are going to be kinases, and the proteins that dephosphorylate them are going to be phosphatases. And that phosphorylation, now you're changing the shape of the protein in response to adding this negative phosphate group. You forgot to pack something in your suitcase and you add something and now your things don't fit this way they did. And so you got to repack everything. And so proteins can undergo conformational changes in responses to the post-translational modifications. You're going to only have a limited amount of protein, and if you want to have more of the protein that you can activate, the way that you do that is through transcription. And so we'll see that some of these pathways are going to lead to short-term responses in terms of activating proteins, but long-term responses in terms of activating that transcription, because it's going to be more efficient that way instead of just dealing at the protein level. If you can actually make more of the protein or stop making the protein, then trying to modify the protein to get it like you had more of it, or to degrade the protein and then keep having to degrade more of the protein as you keep making it. Transcriptional regulation is often going to be happening through things like chromatin remodelers and various proteins that help tell RNA polymerase where to come and what to translate. And we're not going to be talking about those. Just know that they're biochemical bases for these end results that we'll see. But those transcriptional changes have to happen due to things happening in the nucleus. And so our steroid hormones are the only ones that could actually get into the nucleus and do that changing. But we can activate molecules in the cytoplasm that then are able to go into the nucleus and convey the signal. So even though our signal is not coming in the case of most of our signals, we can still relay signals into the nucleus to have those effects on transcription. We're going to focus more, though, at least for now, on what's happening out here, what's happening to the proteins in our cytoplasm and in the membrane. So remember that we could talk about those external ligands, like hormones, as primary messengers. So they're the primary messengers relaying the signal. So they basically are the signal. They're a primary messenger. They're going to bind to this transmembrane protein. They're going to bind to the ectodomain, the part that is outside, cause a change in the endodomain, the part that is inside, and that is then going to cause something to happen. And in the case of our beta adrenergic receptor or our glucagon receptor, some other receptors, what happens is that you generate an intracellular second messenger in response to that primary messenger. So that primary messenger is that small molecule, that ligand, that binds to the receptor outside of the cells. The secondary messengers or second messengers are going to be small molecules that are produced and are released in response to that binding. But the second messengers are acting inside the cells. So the primary messengers bind to the receptor changes shape, it does a bunch of stuff. And one of the things that it can do is increase the production of a secondary messenger, a second messenger, which is a small molecule inside the cells. It's able to relay the signal inside of the cells, even though the primary messenger couldn't get in. And because you're making it inside of the cells, you're then able to amplify the signal by making more of it. And turn off the signal by degrading it. One of the main ones we're going to talk about, basically the one that I care most you know about, is going to be the cyclic AMP or the CAMP, which is produced by adenylyl cyclase in response to binding to the beta adrenergic receptor or the glucagon receptor. There are other types of secondary messengers or second messengers, phospholipid-based ones. So from phospholipase C, you can get IP3 and DAG, and from PI3K, you can get PIP3, as well as calcium. And so calcium is going to be released from intracellular stores, so it's kind of just built up, waiting to go. Whereas these other ones are produced by modifying other molecules. And so the phospholipids in the membrane, well, things are happening in the membrane. So if you chop them up and then phosphorylate them and do things with them, you can make those phospholipids into second messengers. And in the case of our cyclic AMP, we're going to make it by transforming ATP with the help of this adenylyl cyclase enzyme that's going to be activated by this G protein, which is activated by the G protein coupled receptor. And so, again, don't need to memorize anything, don't need to know these various terms and things. Be able to read the diagrams and know cyclic AMP. So let's talk about it. And remember that the pathway we're going to talk about when we get to this G protein is going to be the same in the terms of our glucagon or our epinephrine, or our adrenaline. Remember, epinephrine equals adrenaline. They act through this beta adrenergic receptor. In the case of our glucagon, this part is going to be different, but all this rest would kind of be the same. So let's talk about all this rest of the stuff. So I'm going to give you a really quick overview because I think it's helpful to kind of just see things laid out and see the main steps, and then we'll go into the details of the different steps. First, you have binding of the agonist. So this is our stimulatory ligand. This is going to be our adrenaline. This is going to be our glucagon. It binds to the GPCR, so that receptor protein. Now, that receptor protein is going to activate a G protein. We'll talk in a second about G proteins. They can be monomeric, so single subunit, or multimeric. And so this is going to be a trimeric one we're dealing with. See that it's attached to the membrane through these phospholipid-based linkages. So it can kind of move around this membrane. And this G protein is going to be in its off state when it's bound to GDP. So this protein is in its off state. But then what's going to happen is that binding of the agonist to the GPCR causes that GPCR to associate with the G protein. And it associates in such a way that that G protein is going to swap out a GDP for a GTP, and this is then going to activate the G protein. Once activated, this G protein is then going to associate with adenylate cyclase. Remember, adenylate cyclase was going to be that enzyme that was going to make our second messenger, cyclic AMP. So this GTP-bound G protein, which was activated to make it in that GTP-bound state by this G protein coupled receptor, is now going to be associating with and activating the protein that is going to make our second messenger. We're going to make that second messenger, use it to turn on other proteins down the line, and we're also going to have these other parts of the G protein left over, this beta and the gamma, which are also going to have important roles, but alpha kind of ditches them, and alpha is the one that's going to activate this adenylate cyclase. In order to turn off the signal, well, it turns out that the G protein is actually going to be a GTPase. So it inactivates itself by hydrolyzing that GTP. Once it hydrolyzes that GTP, now it's back to GDP. It associates back with this beta and the gamma, and then you're going to have this inactive form again that is ready to be activated if that agonist is still present. But if the agonist is not present anymore, then you don't have signaling. And we'll see how there's various ways that we could regulate this pathway, and there's various branches off that you could go. But this is the bottom line. You have binding of the agonist to the G protein coupled receptor, activates the G protein. Once activated, the G protein associates with the adenylate cyclase that produces cyclic AMP. And then that cyclic AMP, what it's actually going to do is it's going to promote the breakdown of glycogen to give you glucose. And that is just one of the effects. So now let's go into the details a little more. Starting with what is a G protein? So as I mentioned, these can be monomeric or they can be multimeric. And the multimeric forms are heteromeric. So instead of having multiple copies of this alpha unit, the main unit, the one that actually binds to the GDP and the GTP is called the alpha unit. In the heteromeric ones, what we have is that we have an alpha, a beta, and a gamma. And these are going to be hanging out in the membrane, and they can move around, and they can also associate with the G protein coupled receptor, the GPCR portion. So we've got this alpha, this beta, the gamma, and the alpha is the one that actually has the catalytic activity. It's going to be the one that has the GTPase, the one that can hydrolyze GTP. It's able to switch between the active form in which it's bound to GTP and the inactive form in which it's bound to GDP. It's really confusing because we talk about active and inactive. Basically, if it's on or it's active, then it's bound to GTP, and it's active in terms of it's associating with the proteins that it regulates. Whereas when it's bound to GDP, well, here it's in its inactive state, it's off state, where it's not associating with and activating other proteins. It's confusing because when it like carries out its enzymatic activity, what it actually does is goes from the GTP form, the active form, to the inactive form. And so it's really confusing, but we'll talk mostly in terms of it being active or on when it's just activating something else. It's in that state where it can activate something else, which is that GTP form, and off or inactive when it's in that GDP bound form. It turns out that it binds really tightly to this GDP form. And in order to activate it, therefore, we need to kick out the GDP and so we can bind to GTP. So there is plenty of GTP around. There's a lot more of GTP around than GDP around. So it's easy to pick one up as long as the binding site's open for it. But GDP is going to be bound so tightly in that binding site that you can't pick it up. And so what we need to do is we need to kick out the GDP. And it turns out that there are proteins that allow us to do this, including the GPCR proteins. And so the receptor is actually acting as what we call a GEF. A GEF is going to be a GTP-GDP exchange factor. A GEF turns G proteins on by getting that GDP out. And so our examples, some of them are going to be our GPCR, and we'll see this protein SOS. This is actually going to be involved in insulin signaling. And so, yes, insulin is going to signal through that receptor tyrosine kinase pathway, but that pathway actually involves a G protein. It's not one of those heteromeric ones. It's one of these monomeric ones. And it's a monomeric one called RAS. It's really important. You probably heard about it in the context of cancer, maybe, because it's really important in amplifying signals inside of cells. And so mutations in it can lead to cancer. One of the proteins that regulates RAS is going to be this GEF-SOS. And so we'll talk about that later. But for now, this is just to show you that in response to this GEF binding to this G protein, you get this conformational change that opens up the part where the GDP bounds, allowing for GTP to bind. So that will activate the protein. A GEF will activate it, but we also need to inactivate it. And so we need to have gaps that are regulatory proteins that turn G proteins off by giving GTP a zap. And so basically these gap proteins are going to be GTPase activator proteins. They're activating the GTPase, not activating the G protein. So what they're doing is they're causing the G protein to inactivate itself. They're facilitating the hydrolysis of the GTP. Mutations in these regulatory factors as well can cause cancers. G proteins are not just used for signaling. There are also G proteins involved in translation, so protein making. They act as elongation factors that help the ribosome kick out the used tRNA and put in new ones. But we're going to focus now on the ones involved in signaling. G proteins involved in signaling have to be connected somehow to a receptor. And this might be direct in the case of our GPCRs or indirect in the case of things like when we're looking at SOS and it was kind of several steps removed. In the case of our GPCRs, remember these are going to be our receptor proteins that they have to go through the membrane. And the way they go through the membrane is often in the form of 7 transmembrane domains. And you don't need to know this 7 transmembrane domain term or whatever. I just wanted to introduce you to it because you might see this term 7TM used. And the 7TM is referring to 7 transmembrane domains. And so there's lots of GPCRs, and you might see them referred to as 7TM proteins sometimes. On the outside, the ectodomain is going to bind to that ligand, that primary messenger, remember? And then the endodomain, the part inside, is going to bind to our G protein. And we're going to focus on this GS, this G stimulatory protein that stimulates adenyl cyclase. And adenyl cyclase, remember, was the enzyme that made cyclic AMP. This GS protein is going to be heteromeric and it's heterotrimeric. So it has 3 subunits, these 3 chains, and each of these is going to be different from one another. So we've got an alpha, a beta, and a gamma. We're going to focus mainly on the alpha. I know I feel bad for the beta and the gamma, but for the sake of time, I'm not going to talk too much about what they do. But they do have important functions. But they're not the ones that are actually going to be binding to and exchanging that GDP and GTP. That's going to happen in the alpha. This alpha subunit is actually going to ditch the beta and the gamma when it gets activated. So what happens is that you have this GS and it is bound to GDP, so it's in its off state. And remember, it's bound really, really tightly, so it needs that kick to let it go. And that kick comes from a GEF, which says get out. And the GPCR is going to serve as that GEF. It's going to change shape in response to binding to the ligand. That is then going to cause the GDP to get released. The GDP, once released, now there's a bunch of GTP floating around. The GTP binds to the alpha. And then once bound that, then that activates the G protein. The activated G protein is in this different shape that doesn't want to hang out with the beta and the gamma. So it ditches them. It's got this lipid attachment where it can kind of just diffuse through the membrane. And look who else is hanging out in the membrane? Adenylyl cyclase. It binds to adenylyl cyclase and it activates adenylyl cyclase. Eventually, however, you've got something, maybe you've got a gap come in and give the GTP a zap. And so it actually gives the G protein a zap so that the GTP gets zapped. So this enzyme has intrinsic GTPase activity. It can break down the GTP, but often it's going to need some help from a gap in order to do that. Once it's hydrolyzed that GTP, now it's back to GDP, and then sadly it wanders back, dejected. But it finds its beta and its gamma friends, and now it's ready to do it again. And while he's away, there could be other G proteins that come, and as long as this receptor is still bound to the ligand, it's going to be in that shape that can be activated. And so if you have more G proteins, you can keep activating the G proteins with this same receptor. And so then you need to think about what is the concentration of the ligand out here, and then once it goes away, then this is going to stop. But as long as you keep having ligand here, even when that first copy of the G protein was off and doing its thing, you could keep this signaling going. In terms of the adenylyl cyclase, so what it does is it converts ATP into the cyclic AMP. Now, my undergrad professor, she was like, you do not call it camp. And so I will never call it camp. I'm gonna call it cyclic AMP. The day I die, CAMP is okay, but don't call it camp apparently, or she's gonna get mad at you. No, she was actually really nice. Shout out to Dr. Vidya. But anyway, this adenylyl cyclase is going to cause you to break off this portion and connect this portion to the 3' position. And so you kick off this PPI, and then you are going to form this cyclic structure. And that's going to be catalyzed by this adenylyl cyclase. And then the cyclic AMP, the CAMP, is going to be our second messenger. And then that second messenger is going to activate another protein, protein kinase A or PKA. So a kinase, remember, is going to add phosphate groups, and PKA is going to be a kinase, so it adds phosphate groups to things. But it can only add phosphate groups to things when it's not inhibited. And typically it's got this part that's going to inhibit itself. So it's inhibited in its basic state. It's got these different subunits, and it's got these catalytic subunits to actually do the phosphorylating stuff. And then it's got these regulatory subunits. And the regulatory subunits are kind of jammed into the catalytic subunit, blocking the substrate binding cleft so that you can't have your substrate bind to get phosphorylated. But when cyclic AMP binds, now it's going to bind, cause a conformational change, one of those shape changes in this protein kinase, this PKA, and that is then going to make it so that the catalytic subunits are released and can then go and bind to substrate and phosphorylate things. And this ACAP, this is going to be a scaffolding protein that's kind of going to hold things together. But you've got this PKA with this catalytic and regulatory subunits binding a cyclic AMP. is going to release the catalytic subunits. The catalytic subunits, once released, can then go and phosphorylate things. All that was happening in response to cyclic AMP, which was produced by adenyl cyclase, which was activated by the G protein, which was activated by the G protein coupled receptor. What happens after this? Now you've got a kinase, and a kinase phosphorylates things. Phosphorylation can cause changes that either do things like activate or inactivate proteins. So we've got this kinase that's activated. Now it can go set off a chain reaction by activating other kinases, which can activate other things. And one of the end results is that you get this breakdown of glycogen into glucose. We have this one molecule of epinephrine, say, this one molecule of adrenaline. It binds to that G protein coupled receptor. As long as this ligand is bound, so as long as the adrenaline is bound, even once this G alpha goes away, even once these other subunits go away and they go and they do their stuff, this receptor is still on. It can still activate other ones, still act as a GEF and say, get out GDP. So we can put in GTP, activate the G protein. So you can keep doing that. So these are just some estimations, but you have this one molecule of adrenaline bind, activates about 10 molecules of this G protein, this GS alpha. That is then going to cause adenyl cyclase to be activated, and adenyl cyclase is going to make our second messenger, cyclic AMP. And it's an enzyme, so it can keep doing this over and over and say maybe each is going to make about 20. So overall, we've got about 200 molecules of cyclic AMP made. And we can have those then activate pKa. We get about 100 active pKa maybe. And then that pKa can go and it can phosphorylate phosphorylase B kinase. That can activate phosphorylase B kinase, about 1,000 molecules of it. We're getting a cascading effect. We're getting amplification. What that's going to do then is it's going to take this glycogen phosphorylase B, which is involved in breaking down glycogen, our storage form of sugar. It's going to activate that through phosphorylation, and you get about 10,000 molecules of that active phosphorylase, which is then going to break down glycogen. And then you're going to give glucose 1-phosphate, about 100,000 molecules of that. And when you're in your liver, you can then go and you can take that all the way to glucose that you can release into your bloodstream. And you get about 100,000 molecules of glucose released in response to binding of that one adrenaline molecule. And this is happening in your liver. We're going all the way to glucose, increasing our blood sugar. And so remember, this could happen in response to glucagon as well, because glucagon says glucose is gone. Let's go break down some glycogen and let it out. And then you could have this effect. Or if you just have adrenaline rushing through you, there's a bear coming at you, you need some more glucose in your bloodstream, voila, you've got a way to do that. In your muscles, you don't have this glucose 6-phosphatase. You convert the glucose 1-phosphate to glucose 6-phosphate, which you can use in glycolysis. So you can use it for energy, but unless you have glucose 6-phosphatase, you can't actually make glucose and let it out. So in your muscles, bottom line is you're going to get glucose 6-phosphate, which you can use for glycolysis, but you can't let out. So you get to keep it when you break down glycogen, but your liver is going to break down glycogen to glucose to ship it out. We'll talk much, much more about this when we talk about blood sugar regulation and stuff. But for now, just be able to interpret the diagram, see this green signal that's saying we're activating something. And so we're activating these pathways, follow the arrows, see what happens. That is the key thing about signaling. You might be wondering too, how are we coordinating all of these things? Adapters and scaffolds are going to be your friend. And so here's that ACAP I was talking about. It was kind of holding that pKa in place. And it's also holding other things in place. And some of those other things are going to be things like phosphatases, which can actually remove phosphate groups to undo the effects of phosphorylation. It can associate with adenyl cyclase. It can associate with our GPCR proteins. So everything's kind of held together. And this is great for making sure that the right things can find each other and react. And it's also great for being able to clean up your mess easily. So when you want to remove the signal, desensitize things, you can kind of just whoop, swallow this whole portion of the membrane and then stop the signaling. And so when we talk about a scaffold protein, that would be something like this, where it has binding sites for multiple different proteins. Following arrows was one of the important things that I want you to be able to take away from this lesson. Another is being able to predict the effects of agonists and antagonists by tracking those arrows and seeing what would happen if you have an agonist, which is going to activate, or an antagonist, which is going to inactivate. So let's take an example. What would happen in the presence of an epinephrine agonist or an antagonist? So we've got this epinephrine here. This molecule, it's telling us, is an agonist. So it's going to activate. It's going to have the same effects as the epinephrine. This adrenaline, remember, epinephrine equals adrenaline. Or this propranolol, which is an antagonist. So it's going to have opposing effects. And note that the KDs are listed here. And so we can see that this agonist is going to actually bind more tightly than the epinephrine. And the propranolol is going to bind even more tightly. So really, really low KDs, indicating a really high affinity. Isoproterenol, our agonist, is going to have the same effects as our epinephrine. And so when you think about adrenaline, the effects of the epinephrine can be mimicked by the effects of the isoproterenol. And so you're going to make more glucose. You're going to have all those other effects that adrenaline has if you give them this agonist. And in fact, in medicine, this can be used in surgery and things to keep people's blood pressure from getting too low, because one of the effects of adrenaline is also to raise blood pressure. On the other hand, what if you have blood pressure that was too high? Well, now you'd want an epinephrine antagonist. You'd want something like propranolol. And so propranolol, man, that's a hard word to say. I'm guessing it has some sort of trademark names because it's used in medicine as an example of a beta blocker. And so when you hear about beta blockers, those are used for people with heart disease sometimes or high blood pressure. It's going to act as an antagonist of this adrenaline, of this epinephrine, and therefore do things like lower the blood pressure and lower the amount of glucose that gets made and released. In addition to predicting the effects of agonists and antagonists, you should be able to think about what would happen in the case of a mutation. Mutations in these G proteins and proteins involved in various signaling pathways are very common in cancer. If you have activating mutations in G alpha S, say, those are going to lead to a continuously elevated levels of cyclic AMP. And that's found in about 40% of adenomas. Conversely, inactivating mutations can cause people to be unresponsive to hormones that act through the cyclic AMP. How could we stop the signaling in the absence of mutations? Well, one player is insulin. So remember, insulin and glucagon were going to have opposing effects. Insulin was going to say, let the glucose in, whereas glucagon was going to say glucose was gone. And so insulin is going to have opposing effects to the glucagon. And so we can imagine that it's going to have some sort of negative effect on our glucagon signaling. And we'll see that when we talk about glycogen regulation, insulin is actually going to be able to help control the glycogen synthesis and the glycogen breakdown. And so insulin is going to promote the synthesis and inhibit the breakdown. And one of the ways that it's going to do this is by controlling a phosphatase that can remove some of those activating phosphate groups that were put on by this PKA. And so insulin is going to work through PP1, which is going to inhibit the effects of the PKA. So again, more on this later. But for now, just get familiar with seeing these sorts of diagrams where we're seeing that things are getting inhibited and things are getting activated. And these are going to have crosstalk between the different pathways. Focus on being able to follow the arrows and see the end results. So we were just talking about our glucagon and our epinephrine that were acting through this GPCR, which was activating the GS alpha, which activated adenylate cyclase, which produced cyclic AMP, which activated PKA, which activated phosphorylase kinase, which activated glycogen phosphorylase. And glycogen phosphorylase is the enzyme that is going to be catalyzing the breakdown of glycogen, releasing units of glucose 1-phosphate. And so when you see an enzyme name shown above the arrows, it's saying it's catalyzing this reaction. And so glycogen phosphorylase breaks down glycogen, glycogen synthase makes glycogen, and glycogen phosphorylase is activated by phosphorylase kinase, which is activated by PKA, which is activated by cyclic AMP, which is activated by adenylate cyclase, which is activated by GS-alpha, which is activated by G protein coupled receptors, which were activated by epinephrine or glucagon. Whew, see? Yeah, it's a lot, but if you follow the arrows, you're good. Also, just I know I said I wasn't going to talk about beta and the gamma, but I felt bad for them. And so, one last note about them, they actually are important. So, once the alpha ditches them, they can do things like help promote the internalization of that GPCR, the actual receptor, because the receptor can keep activating things as long as it's on the surface. The beta and the gamma can help promote the kind of internalization of the G protein coupled receptor so that you don't have that response just going all the time. You're able to kind of desensitize. And this is one of the ways you can desensitize is with the help of the beta and the gamma. That would stop it at the source, but you still have all that stuff activated further on. And remember, the cyclic AMP was going to keep amplifying our signal. And so we can remove the cyclic AMP. We have an enzyme called cyclic nucleotide phosphodiesterase, or sometimes you just see it referred to as phosphodiesterase or nucleotide phosphodiesterase. It is going to act as a hydrolase, and so it takes water and it uses that water to break down this cyclic AMP, and it breaks it down into AMP. So remember, we started with ATP. Adenylyl cyclase took it to cyclic AMP and released this PPI. So now we only have one phosphate group. So if we uncyclize it, now we get the uncyclase form of this, and that uncyclase form is going to be your adenosine 5' monophosphate, or AMP. This nucleotide phosphodiesterase is inhibited by caffeine. And so if you have caffeine, you increase your levels of cyclic AMP. And that's just one of the effects of caffeine. It also does other stuff. We can also regulate these G protein coupled receptor pathways with our GEFs, which remember say, get out GDP, so that you can put in GTP. So your GEFs turn G proteins on, but then you have your GAPs, which turn G proteins off by giving GTP a zap, helping the G protein hydrolyze the GTP, inactivating it back to its GDP bound state. And this was just one of the pathways that we could take through that G protein coupled receptor, was this adenylyl cyclase PKA path. And in addition to all that effects on the glycogen, it affects gene regulatory proteins as well, and many other target proteins. Some GPCRs are going to signal via different second messengers. And so you'll see that here we actually have this GQ subunit. There's also GI subunits, various other subunits than the GS. We're just going to focus on the GS, but you should be able to kind of follow arrows and things and see what's going on. One of the things that's going to be going on is that some of them are going to signal through this phospholipase C based pathway. Phospholipase C is going to be chopping up a phospholipid in the membrane to create these secondary messengers, DAG and IP3, basically the head group and the tail. And what those are going to do is they're going to have effects intracellularly. And so all of that is happening in response to a hormone binding to a receptor. You don't need to know these pathways, but I just left this in here in case you wanted to know. So the signal binds to the receptor, it activates this GQ. GQ then activates phospholipase C. Phospholipase C chops off the head of this PIP2. That gives you DAG and it gives you IP3. IP3 is then going to this head group that you just chopped off is going to dissociate, and it goes and it opens up a calcium channel. The calcium channel is then going to let out calcium, which is then going to act inside the cells as the second messenger. And one of the things that's going to do is it's going to activate this protein kinase C. The protein kinase C can then go and act as a kinase and phosphorylate things. What about the DAG? Well, the DAG was that tail portion, so it's still stuck in the membrane. What it's going to do, though, is it's going to be needed in order to activate protein kinase C. So you've got to have these kind of dual signals activating this. And then a lot of other stuff happens because you've activated a kinase, and a kinase can phosphorylate things, which can cause them to change shape. It might activate another kinase, which activates another kinase, which activates another kinase, and this is kind of going to get us into what we're going to see with our receptor tyrosine kinase. One of the ways that calcium affects the proteins is through this calmodulin. And this calmodulin protein, it binds to calcium and it majorly changes shape. And then calmodulin acts with other proteins and causes them to change shape, and you get these signaling effects. So there's lots of different proteins that act through various pathways that are associated with these second messengers. And we're not going to go into them. But PKA is the one that's going through that G protein coupled receptor with the stimulatory subunit, that GS. This was happening through like the GQ subunit, which you don't need to worry about. But GS was going through PKA. Then this other pathway was going through PKC. And then there'll be a PKB, don't worry. And PKB is also going to be known as AKT. There's a lot of abbreviations. And there's a lot of G proteins, and they respond to different types of signals. Some of them are actually going to be inhibitory. So if you see a GI that inhibits adenylyl cyclase, whereas a GS was going to activate it. So remember we talked about how adrenaline kind of raised your blood pressure? Well, it was doing that by activating this adenylyl cyclase pathway through this GS. And vasopressin, which is going to lower the blood pressure, acts through this GI, this inhibitory subunit, which lowers the production of cyclic AMP and therefore the PKA activity. And there are lots of different G proteins that respond to lots of different types of signals. There are also lots of receptor enzyme signal transduction pathways. And so these are typically RTKs, or receptor tyrosine kinases, and we're going to talk about the insulin receptor, which is a classic example. RTKs act as dimers, but not all of them are constitutive dimers. The insulin receptor is going to be a constitutive dimer, and so it's always a dimer, whether or not it's active. But other RTKs are going to normally be monomers. And so you can see this GF, when you see GF, think growth factor. So there's a lot of different growth factors that act through RTKs. And these are often going to be monomeric. And then in response to, say, a ligand binding, they're going to come together. In the case of our insulin receptor, it's always a dimer, but it's in an inactive conformation until the ligand, so in this case, insulin, binds. That causes the kind of two legs of the receptor to come together and phosphorylate one another. So we call this autophosphorylation when something phosphorylates itself, but here it's kind of cross-phosphorylation between the different chains. So you have these catalytic subunits inside of the cell in this endodomain, and you've got this receptor binding portion and this exodomain, and then you've got this transmembrane portion that's connecting the two. So the insulin binds in the ectodomain, causes a change in the transmembrane portion, which causes a change in the endodomain, which causes the phosphorylation of this kinase. When we talk about a tyrosine kinase, that's talking about it phosphorylates tyrosine residues, not that it's using a tyrosine residue. And so it actually phosphorylates tyrosine residues on various substrates. And this phosphorylation is going to open up kind of the substrate binding portion so that things with tyrosines can go in there and be phosphorylated. And once you have those phosphorylated tyrosines, then they can bind to other things. And so there are phosphate binding domains on various proteins. Some of the main ones are going to be SH2 domains. Those are going to bind to phosphorylated tyrosine residues. And so you can have SH2 domains on various proteins that are then going to be able to bind to whatever was phosphorylated by that RTK. You're going to have proteins that have a lot of different domains hooked up. And these are going to be great for scaffolding and things. And so you'll see scaffolding proteins are often going to have these SH2 domains, which allow them to bind to phosphorylated residues. You also see SH3 and pH domains, and those are going to bind to the phospholipid PIP3. I'd like you to know SH2 domain, but you don't need to worry about SH3 or pH. But just know SH2 domains are going to bind to phosphorylated tyrosine residues, and those phosphorylated tyrosine residues are often going to be coming based on phosphorylation from a receptor tyrosine kinase, something like our insulin receptor. So let's talk about a couple of the main ways the insulin can signal through the insulin receptor. And so we have our insulin receptor. Sometimes you'll abbreviate this INSR. Remember, it's a dimer, but it's not active until it gets phosphorylated. It phosphorylates itself in response to insulin. And so insulin binds, and it causes the dimer to autophosphorylate. And then that is then going to activate the kinase. Remember, this is a kinase, the receptor tyrosine kinase. So in the G protein coupled receptor, we didn't have an enzyme in our membrane, we just had that G protein. But here we actually have an enzyme, we've got a kinase. We're activating a kinase directly when we have that messenger bind, when we have that insulin bind here. So we've activated a kinase, and a kinase now phosphorylates things. The kinase is going to phosphorylate this protein called IRS1, insulin receptor substrate 1. IRS1, once phosphorylated by this kinase, is then going to go and it's going to interact with molecules that lead to the activation of RAS. So remember, we saw RAS earlier. RAS was that G protein that was monomeric, and so it was a single subunit. It basically just had that alpha subunit. And so what it can do is it can hydrolyze GTP. And what it can do is if it has GDP kicked out, it can bind to GTP. And remember that the things that got the GDP to get kicked out were our GEFs. So our GEFs activate our G proteins. And this is that GEF I was telling you about before, that SOS. SOS is going to be a GEF that's going to activate the G protein. But SOS itself needs to get activated, and it gets activated when this GRB2 gets activated. And so this GRB2 has one of those SH2 domains that binds to a phosphate group, and it binds to a phosphate group on this IRS1 protein. So this IRS1 protein, now that it's phosphorylated, it associates with GRB2. GRB2 associates with and activates SOS. SOS then activates RAS, and RAS is then going to be able to activate RAF. RAF is a kinase. Kinase phosphorylates things. We're going to get a phosphorylation cascade. And so we get this RAF is going to activate MEK. MEK is going to activate ERK. ERK is going to go into the nucleus and affect transcription. And so you have this pathway where you're able to take that signal of insulin, and now you're going to be able to control the genes involved in things with glucose regulation, in things with other types of metabolism, other types of processes, other types of growth processes, maybe all in response to that insulin binding. So again, we have insulin binding to the receptor. That causes the receptor to autophosphorylate itself. That activates the receptor. The receptor is a kinase. So it phosphorylates tyrosine residues in IRS1. IRS1, once phosphorylated, associates with this adapter protein GRB2. GRB2 associates with SOS. SOS is a GEF. It says get out GDP so that our G protein can get activated and bind GTP. Once activated, our G protein, in this case RAS, is able to activate other things. It activates this protein RAF. RAF is a kinase. It can activate other kinases by phosphorylating them. It phosphorylates the kinase MEK, which activates MEK. MEK then phosphorylates and activates ERK. And then ERK then goes into the nucleus and affects transcription. So RAS, RAF, MEK, ERK. And so you don't need to memorize that. I don't know why, it's just stuck in my head from undergrad. And those are just examples of larger families. And so this is just for giggles because these things get a little ridiculous. So ERK is in the MAPK family, myogen activated protein kinase. But it got activated by MEK. And so we have this MAPK protein that's activated by a kinase. So now we get this MAPKK, this MAP kinase. But then that was activated by another kinase. So now we need a MAPKKK. We need a MAP kinase family. And so we get these kind of ridiculously long names, and then we have these short names. So that's that. And this is often going to be facilitated by having those scaffold proteins that are then able to bind to these various substrates. And often they're going to be binding through things like SH2 domains. All giggles aside, mutations in these G proteins like RAS can cause cancer. And RAS doesn't just associate with RAF, it also associates with a bunch of other proteins. And so you can have mutations in RAS that are again going to influence a lot of different pathways and cause a lot of problems. One of these other pathways is going to be this phospholipase C. Phospholipase C is another way that insulin can signal through. Phospholipase C, remember, was that one that was then going to give us that IP3, that calcium, that calmodulin, all that PKC stuff. That was happening through that phospholipase C, and that phospholipase C was going to be activated by this insulin signaling, as well as by the G protein coupled receptor signaling. So we have crosstalk between the different pathways that are kind of converging on similar secondary messengers in some cases. So let's talk real quick about this phospholipase C pathway, because it's going to lead to an important effect. And it's not that I care you know this pathway, but I want you to know the end result. That in the muscles, the heart, and the adipocytes, so the fat cells, insulin also signals through PI3K to lead to the movement of GLUT4, which is a glucose transporter, to the cell surface to allow glucose in response to insulin. And so PI3K is going to be a kinase. So when you see K, it's a good guess, it's a kinase. And so it is going to be this kinase that what it does is it takes PIP2, which is a phospholipid, and it changes PIP2, it phosphorylates it to give you PIP3. So PI3K takes PIP2, makes PIP3. It phosphorylates PIP2. It's going to only phosphorylate PIP2, however, in response to binding to phosphorylated IRS1. And IRS1 was that same protein that we were looking at before that was phosphorylated by the insulin's receptor protein. So we have the insulin bind. Our dimer autophosphorylates. That's going to phosphorylate IRS1. Before we saw how IRS1 was then going to associate with BREB and associate with SOS, which was going to activate RAS, here we're going to see that instead it's going to associate with this PI3K. Now PI3K, that was going to phosphorylate our PIP2. PIP2, once phosphorylated, we call it PIP3. Now PIP3 is going to activate PKB. And PKB is also known as AKT. One of the effects that it's going to have is through a couple proteins called RAB and REC, it's going to lead to the incorporation of glucose transporters into the membrane. So these glucose transporters, these GLUT4 transporters, are pre-made and they're held in these little vesicles. So they're already in membranes, they're already in the cells, they're just kind of in these cellular bubbles. And then what happens is in response to the signaling, you're going to get them incorporated into the membrane. So when you have the insulin, now you can put those receptors in the membrane in order to bring glucose in. But this is only happening in certain tissues like your muscles, your heart, and your fat cells. PKB also inactivates GSK3, which was then preventing the inactivation of glycogen synthase. But the key thing here is we have that this PKB, this AKT-based signaling through the insulin receptor is able to lead to the bringing of the glucose transporters into the membrane of the cell, which will allow you to take glucose in. Because remember, insulin says let glucose in. A lot of them are going to be important for controlling cell division. So these cyclic dependent protein kinases that are made in response to growth factor-based signaling through RTKs. So see our MAPK cascade and stuff. Those are able to kind of control the cell cycle and make it so that the cells divide and divide and divide, or don't divide and don't divide and don't divide. And so when mutations cause these to divide and divide and divide, they can lead to cancer. And so some of these you might have actually heard about, things like HER2, and HER2 is often involved in breast cancer. And so HER2 is overexpressed in some breast cancers, and it is an example of a RTK. And it's also an example of an RTK that doesn't need a ligand. And so it's always going to be active. And normally it can pair up with other RTK monomers. But if you have a lot of it, because normally there's not that much of it, but if you have a lot of it, if it's overexpressed like in a cancer, then you get this sort of constitutive signaling. You have excessive signaling because you're not relying on there being a ligand present. In cancers that overexpress HER2, one of the treatments is Herceptin, which is an antibody that is a little protein that will bind to that HER2 receptor and act as an antagonist, prevent it from getting activated, and therefore prevent this excessive signaling. There's a lot of crosstalk between pathways. We saw some examples. There's also examples where various pathways can inhibit other pathways. and we're not going to go into that. And much of it's going to be mediated by phosphorylation-dependent binding. So our kinases, the enzymes that add phosphate groups, and the phosphatases, the enzymes that remove phosphate groups, are crucial to biosignaling. Speaking of biosignaling, let's just review those four main types. We have our G protein coupled receptor. We have the ligand bind to this G protein coupled receptor in the membrane. That receptor is associated with the G protein. That G protein, once activated, activates other proteins. Our classic example is our beta adrenergic receptor and our glucagon receptor, which remember are both act through that PKA and cyclic AMP pathway. Receptor enzymes, so our receptor tyrosine kinase, for example, like our insulin receptor, those bind to a ligand, which causes the dimer to activate itself, or it causes the monomers to come together to a dimer that activates them, and then it carries out phosphorylation of intracellular substrates, often through signaling cascades. And these can have effects like leading to the transcription of various genes. In these cases, our primary messenger didn't get into the cells. The only case where the primary messenger gets into the cells is actually going to be in the case of our steroid hormones, where we have nuclear receptors. The steroid comes in, it binds to a receptor protein that's actually in the nucleus and is able to affect transcription of genes and therefore production of proteins. And then finally, we had our gated ion channels. And so our gated ion channels, those are just facilitated diffusion channels, and they open up in response to the binding of a ligand, and then they let things out. And we actually saw an example when we were looking at calcium. And so basically, calcium had a ligand gated channel, but it was in an internal vesicle. And so we were releasing calcium into the cells, and as opposed to letting ions go in the cells. But in those cases, in all these cases of these gated ion channels for membranes, the ligand itself is not coming in. It's the ions that get to come in response to the ligand binding to that channel and opening it up. So voila, signal transduction.